Hello and welcome all to my YouTube channel. Today I am here with our next topic, cell cycle. But before discussing cell cycle, let us discuss about basic terms related with cell cycle and cell division. As you all know, in all living organisms, growth and reproduction, they are the two characteristics of all living cells or organisms which are important for their survival as well as for the continuity of race. And these growth and reproduction, they are interlinked with each other to bring about major changes in terms of cell division and continuity of a race or forming a cell lineage. In reproduction, a parental cell type divides and it gives rise to daughter cells through a process. That process is called as cell division, and basically the result of this reproduction and cell division phenomena is formation of daughter cells. But the formation of daughter cells from parental cells through cell division, it is not a unilateral process. It is also accompanied by a continuous process of increase in terms of cytoplasm, that is growth. So growth, it is basically a continuous process where there is increase in terms of cytoplasmic content, and it also involves synthesis of genetic material, that is DNA synthesis, along with synthesis and accumulation of other types of carbohydrates, lipids and proteins. And this overall process of growth, it is linked with cell division, and subsequently daughter cells, they are formed, and these daughter cells again follow same process and they grow, then they reproduce their different cell lineages or daughter cells based on the same statements, that is the characteristic features of living cells or organisms, growth and reproduction. They were also assessed by many scientists over the time, and one of them was Rudolf Virchow. He gave a statement, and that statement is, as you all know, it is omnis cellula e cellula, and it means new cells arises by the division of pre-existing cells, that is new cells arises by the division of pre-existing cells, and this is also called as cell lineage theory. It is also called as cell lineage theory, omnis cellula e cellula. That means new cells arises by the division of pre-existing cells, and this statement, it was given by Rudolf Virchow, and it is also known as cell lineage theory. And as a whole, it can be compiled or concluded as that large number of cycles of growth and divisions allow a single cell to form structures consisting of millions of cells, that is cell lineage, daughter cells. Now there are large number of factors which are responsible for cell division, and one of the factor is surface to volume ratio, and it is considered to be the major cause of cell division. Usually what happens when a cell grows in size, its volume increases but its surface remains same. Now this can create a problem to the cell, as its volume is ever increasing but surface it is same or it remains constant. So this can create insufficient surface to hold increasing cytoplasmic content, and ultimately cell is triggered to follow cell division. And second factor is mitogens. They are signaling molecules which are usually synthesized and secreted by cell organelles into the cytoplasm, and they form a gradient that ultimately form a conducive environment to perform cell division. So coming to our main topic, that is cell cycle. The definition of cell cycle, it is an ordered series of events or sequence of events by which cell duplicates its genome along with its cytoplasmic constituents and divides it into two daughter cells. That is called a cell cycle. It is a sequence of events, and here duplication of genome as well as cytoplasmic constituents that involve cellular cytoplasmic organelles, and ultimately two daughter cells they are formed. And the time period which is required to complete one cell cycle, that is from beginning of one cell division to the beginning of next cell division, this time period, it is called as generation time. It is called as generation time. That is a period between beginning of one cell division to the beginning of next cell division, that include a complete cell cycle. So cell cycle, it is divisible into different phases, and here two major phases of cell cycle are interphase and M phase. Interphase is the phase of cell cycle where a cell usually spent its 95% of life cycle, and remaining 5% it is it involve your M phase. And interphase, it is further divisible into three different phases, that is G1 or gap 1 phase, second one is S or synthesis phase, third one is G2 or gap 2 phase. It involve growth, it also involve growth, it is also called as growth 2 phase, and it is synthesis phase. While M phase, it is also called as mitotic phase, and broadly it is further divisible into two phases, that is first one is the division of nucleus, that is called as karyokinesis, that is division of nucleus, and second one is cytokinesis, that is division of cytoplasm. And in mitotic phase, these karyokinesis and cytokinesis, they are further divisible, they are further studied under different phases of mitosis that include prophase, metaphase, anaphase, and telophase. From prophase, metaphase, anaphase, and to early telophase, here karyokinesis occur, and in telophase, cytokinesis, that is division of cytoplasm, and ultimately daughter cells are formed. So different phases of cell cycle that include G1, S, G2 and M phase, they have different time duration that varies from species to species and even within same species individuals, from tissues to tissues, organs to organ, and they have differential durations. And on an average, that is time duration, that is the time required for cell cycle to complete its one division from parental to daughter cells, and it is having on an average in case of humans around 24 hours time to complete one cell cycle. It take around this much time, and in yeast cells, in yeast, approximately 90 minutes they are sufficient to complete one cell cycle. So next, coming to our main phases of cell cycle, that is interphase. Let us discuss about some basic features of interphase and how it is different from M phase. In interphase, there occur no cell division. First point, there is no cell division, but here cell undergo many catabolic and anabolic changes. There are number of catabolic and anabolic changes, and first no cell division, but catabolic and anabolic changes are there, and they have major role because these changes they make cell fit for division, that is fit for division. So it is clear from the statement interphase, it do not involve any cell division, that is daughter cells are not formed, but here number of catabolic and anabolic changes they occur, and these helps in making the cells fit for cell division, means they have all credentials and have fulfilled whole its requirement to carry out M phase or mitotic phase. And on the other hand, this M phase, here karyokinesis is followed by, first one is karyokinesis is followed by cytokinesis, but here occur no replication, no DNA replication. And interphase, here these anabolic processes and catabolic processes they involve DNA synthesis by DNA replication, but here in M phase there occur no DNA replication, and here just the already synthesized DNA, it is separated into two daughter cells, and it is regulated, both these interphase and M phase they are highly regulated by number of signaling molecules, and these are called as cyclins and cyclin dependent kinases. Cyclin dependent kinases, and these cyclin dependent kinases they are also called as CDKs. They form complex, and this complex then phosphorylates specific target substrates, and cell division is carried forward. Now, as I have earlier discussed with you that cell cycle involve two phases, and these are interphase and M phase, and interphase is the major phase where cell cycle spend 95% of its duration, and here it has three different phases. So this is M phase and this is interphase. It is as a whole, this one here, it is interphase, and interphase is further divisible into G1 phase, S phase, and G2 phase. G1 phase on an average where interphase has a duration around interphase having duration around 10 to 20 hours. Then G1 phase, it took around 6 to 8 hours, and S phase it took around 5 to 7 hours, and G2 phase took around 2 to 5 hours, while M phase it is completed within 30 to 3 hours duration. So here 95% of cell cycle it is by your interphase, and that is divisible into G1, S and G2 phase, while 5% or less than 5% it is by M phase, and it has differential duration among different species and within same individuals or different cell types within same individual. So coming to the main events that occur during G1, S, G2 and M phase. In G1 phase, that is G1 phase, it is having high metabolic rate, and due to this there is cell increases in size, and there also occur doubling of cellular organelles, but all organelles they are not duplicated or doubled here, and an exception is centrioles, they are not duplicated here. And apart from this G1 phase involve the synthesis and storage of number of NTPs, that is nucleotide triphosphates, and deoxynucleotide triphosphates, which are required for the synthesis of DNA and RNA during S phase, and also various enzymes or regulatory factors which are required for synthesis of new DNA molecules along with different RNA types. And here this is the main phase of interphase where cell attain maximum size or maximum growth it occurs. In S phase there occur replication of nuclear DNA, so this is nuclear DNA, not the cytoplasmic DNA. The cytoplasmic DNA which is found in mitochondria and chloroplast, they are replicated in any phase of interphase. They can be replicated in G1 phase, they can be replicated in S phase and G2 phase, but the nuclear DNA they are always and always replicated in S phase of cell cycle. And here synthesis of, second point is synthesis of histone proteins. Also occur. And third point, this is second point, and third point here, centrioles, they are also duplicated. They are also duplicated in the cytoplasm. So it is clear from this here, the replication of just nuclear DNA and the cytoplasmic DNA which are found in mitochondria and chloroplast, they are replicated either in G1, S or G2 phase, but nuclear DNA, they are replicated just in S phase. And here S phase also involve the synthesis of histone proteins, and centrioles, they are also duplicated in the cytoplasm. They are not duplicated in G1 phase along with your other cellular organelles, and here they are duplicated. And third phase is G2 phase, and this phase is also concerned with post DNA synthesis growth, that is post DNA synthesis growth, and this prepares cells for division, as it involves the division of more and more mitochondria so that they can immediately produce large number of ATP molecules. So it means there is increased number of mitochondria, and they provide basically ATP molecules during spindle formation and during mitotic phase. And also here occurs synthesis of mitogens which are required for subsequent phases of cell cycle. And here major event, that is synthesis of tubulin proteins, and these tubulin proteins, as you all know, they are required for formation of spindle fibers. So this is the main function of G2 phase. Here number of mitochondria are formed through binary fission, and they are involved in synthesis of more and more ATP molecules, which ultimately helps in mitotic phase to bring about karyokinesis as well as cytokinesis. And second, number of mitogens, they are also stored here in the cytoplasm. And third, more and more tubulin proteins, which are the basic monomeric units of spindle fibers and microtubules, that ultimately helps in formation of mitotic apparatus and separation of sister chromatids between two daughter cells. And M phase, it involves different subphases, and here first phase is prophase, second phase is metaphase, third phase is anaphase, and last phase is telophase. So there are four different phases. These are subphases of mitotic phase: prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and early telophase, they involve an event, that is major event of M phase. It is karyokinesis, that is division of nucleus, and subsequently there occur cytokinesis, that is division of nucleus, and two daughter cells are formed from one parent cell. If I draw the general diagrams of these different cells and at their different phases of a cell cycle, here for example there are cells, daughter cells, and a cell enter into G1 phase. So this is G1 phase, and it is having one and having other one chromosome set, that is this orange colored, it depict maternal chromosomal set, and this blue color depict paternal chromosomal set. So they are diploid, and they have two chromosome sets, diploid and two chromosome set. Now this G1 phase, here cell grow in size and cell attain maximum size or maximum growth, not maximum size, maximum growth among all phases of cell cycle. Here the cell attain maximum size and growth, and chromosome number they remain same, and cell is diploid and chromosome they are two sets. So basically they are homologous pair of chromosomes. Now when cell enter into next phase, that is S phase or synthetic phase, here there occur no growth in size of the cell, but here replication of DNA occur, and these cells they are having four chromosomes, four homologous chromosomes, but these chromosomes they are not separated, and these cells are also diploid one. Cells are diploid and they have DNA content found on sister chromatids of these homologous chromosome pairs, which are the result of these maternal and paternal chromosomes. As these chromosomes are held at their kinetochore region, so here just DNA content doubles and chromosome number it remains same. As a whole the cells, that's why they are diploid. They are not called as tetraploid or 4n, but here chromosome number remains same, but DNA content within same chromosome it is doubled, so they are also diploid. And this is S phase, and now in the next phase cell grow in size as it accumulate other number of mitochondria, cellular organelles, and here chromosome number that is also same, they are diploid, and here chromosome they are also 4C. So this 4C and 2C they just represent the DNA content within same chromosome. So here DNA content is doubled and here DNA content is half, because there is no replication at G1, but after G1 in S phase there occur replication and chromosome they have double DNA content and they are joined at their kinetochore region. So sister chromatids they are not separate chromosomes, they are still single chromosome. And in M phase cell undergo first karyokinesis and then cytokinesis, and two cells they are formed. So this is one cell, this is other cell. So here it is having one copy of this chromosome and other chromosome, and cells they are also called as diploid, and this is also diploid cell. That is the result of cell division, and these are also called as daughter cells. So they are the result of this cell cycle. And here diploid cell first replicates its DNA content and then through mitosis separate it into two daughter cells. Some cells in the adult animals, they do not appear to exhibit cell division. For example, there are certain cells, for example in cardiac muscles, even other skeletal muscles. Then RBCs, nerve cells, they lose their division and they are terminally differentiated cells. When cell cycle is accomplished, these cells they are formed, but further they do not undergo repeated cell cycle or subsequent phases of cell cycle. And here what happen, during G1 phase they become differentiated. They are differentiated here and they do not undergo cell division. They are metabolically active. Metabolically they are active, but they do not proliferate or divide. So there is no proliferation or division. And such cells which enter into this metabolically active but non-proliferative phase, so this stage it is also called as G0 phase. It is also called as G0 phase, or in other terms it is also called as inactive stage. And this inactive phase it is also termed as I am writing the term for this is at here, it is also called as quiescent, quiescent stage, that is G0 phase. Here cell escape subsequent phases of cell cycle and they are differentiated from G1 phase, and they are metabolically active but they do not undergo proliferation phase. An example is cardiac muscles, skeletal muscles, RBCs, nerve cells, and these are cell types which remain in G0 phase, quiescent stage. So finally is just one more revision. G1 phase it is also called as pre-DNA synthesis phase, or it is also a time interval between mitosis and initiation of DNA replication. And second is synthesis phase. S phase is synthesis phase, because here replication of nuclear DNA is the major event, and along with this histone proteins they are also synthesized. And G2 phase it is also called as post DNA synthesis phase. This is pre DNA synthesis phase and this is post DNA synthesis phase. If you like my video, you can share it, comment on it, and can subscribe my YouTube channel for many more such videos. Thank you.